Well, welcome everyone to this afternoon's Soil Wealth and Integrated Crop Protection webinar. My name is Carl Larson from RMCG, and today we're going to be talking about area-wide management of diseases, and really happy to be joined by some leading researchers from the Queensland Department of Agriculture and Fisheries to take us through some of the recent research that they've completed for Hort Innovation. I'd like to acknowledge, firstly, our funding body, Hort Innovation, and also the Vegetable Levy and our partners at Applied Horticultural Research as well. So what are we going to cover today? Firstly, we're going to be covering the broader area-wide management project and some of the main outcomes before we delve into a couple of other areas around virus disease outbreaks and weather, as well as pectobacterium disease in zucchini. We've also made sure on our agenda today, within the hour, we've got plenty of time for your questions and a chance to engage with our presenters. And if you haven't joined one of our sessions in the past, on your GoToWebinar control panel, you'll see other attendees and who's on the line, and we've got 50 people joining us from around the country and overseas today, which is great. There's also a pane there for questions, so at any time throughout the session, feel free to type them in and they'll come through to me and I can pop them to our presenters. There's also a panel there for handouts to take away and quickly just click and download rather than searching around the web for some of the relevant material we're going to cover today as well. So before we do get stuck into things and introduce our presenters for today, just a little bit about the Soil Wealth and ICP project. So this is an extension project really focused on improving soil management and plant health around the country and putting some of the great research into practice on farms with growers and agronomists. So we've worked directly with industry over a couple of phases now of the project for the past 8 years, and really trying to be responsive to industry needs. So we've got a network of demonstration sites around the country. We run events similar to these webinars, but also farm walks and masterclasses, and have a number of resources available both on the website and through some of the social media channels as well. So our speakers today, we're really lucky to be joined by Cherie Gambley, Paul Campbell and Rebecca Roach, and we'll be starting off with Cherie talking through some of the main outcomes from the area-wide management project, which has been running over the past years. So Cherie, I might hand over to you and share your screen. So that should be coming through now. Welcome and thanks very much.
Thanks, Carl, and thanks for the opportunity to talk today. So I'm just going to give you a quick overview of what we've been doing for the last 4 years. So let's start. This is a really big project. Sorry, I'm just moving things so I can see the screen. There was about it was a really big project. It was worth about $10 million in cash, and that was a 50-50 co-investment through the Veggie Levy Fund and through our agencies like DAF. And then there was another $6 million in-kind contributions to the project. So it's been a 4-year project, and it's national, and we've enjoyed working together and working with industry over the past 4 years, and have delivered quite a lot of things. We have Collaborative Link New South Wales. DPI has a similar project, and we work really closely with Tony Chapman in that project. Okay, so it's a really big project. So there was a really big project team. Queensland as a lead agency, we had a significant number of people working on this, as did some of the other agencies. Not everybody worked for the whole 4 years on the project. People came and went. And we also had quite a few private research providers, which were fantastic to link with. It gave us more of an industry focus in some areas. So we had Anthony Rice up at the Granite Belt doing some monitoring, particularly for phytoplasma and leafhoppers. Len Tesoriero from New South Wales, who helps pretty much with everything on the project. Similarly, John Fletcher, who's a virology consultant, who's living in Australia, moved over here from NZ. Chris Tempson up in Bundaberg has been great in running a lot of our field trials that we've done in the project. And similarly with Chris Monsour up in Bowen. And then we also had Stuart Grigg down in Victoria helping with some monitoring of thrips and tospoviruses in lettuce. The project also had 4 PhD students, which are all finishing up in the next month or two, and they've done a great job. So I'd just also like to acknowledge industry. Without industry's help, we can't really do this project. So part of the project, we're doing disease surveys nationally, and so we've been to a lot of people and visited a lot of people, and we thank them for their time. But in addition to the disease surveys, we also had a lot of people where we worked directly on their properties, collecting data and working with them to solve some of the disease problems. And this list here just lists the ones that I've been involved with in Queensland. And like I say, this was a national project, so there'd be lists like this from all around the country where our interstate collaborators have been working closely with industry in their states. Also like to have an acknowledgement for Ausveg. Ausveg have been fantastic supporters of this project, particularly Callum, Maddie, Zali and Danielle, and we've really enjoyed working with them. We've done a lot of workshops together and surveys together, and it's been really good to have their perspective. So it's a big project, and so the scope of the project's quite big as well. We covered basically viruses, bacteria and phytoplasmas that affect vegetables all across Australia, all the insect vectors that transmit these. We've done some biosecurity preparedness through exotic viruses and bacteria. So we've done 3 viruses and 3 bacteria, and they'll be available through Plant Health Australia. We've developed a whole heap of low-tech and fast diagnostics. So when you send us samples, we can do them faster, or some of them you can do yourself. We've been doing a lot of monitoring of insect, weather and disease, and Paul's going to give some examples on how weather impacts virus disease outbreaks. We've done a review of seed as an entry pathway, and we've had 2 workshops on that. And Hort Innovation's commissioned a RFP for further work looking at seed through diagnostics to help harmonise and improve efficiencies of seed testing. We had, in terms of capacity building, we definitely have rebuilt the pathology resource for the vegetable industry in relation to virology and bacteriology. There was a significant hiatus for probably nearly 10 years, and so it's nice to have that national reconnection of the virologists and the bacteriologists with the vegetable industry. And we've put through 4 PhD students. We've also provided free diagnostics for the industry, but sadly that comes to an end at the end of the project. So what resources are there for you? So the big ones for the project are 2 disease guides. One is Understanding and Managing Bacterial Diseases, which is available now as a hard copy. And also we're just finalising this month one for virus diseases. So if you want to get a hard copy of these, please register today. There'll be an electronic downloadable version as well through Hort Innovation and Soil Wealth when we've finished finalising them. We've also developed up a priority list of endemic and exotic pathogens that affect vegetables. This is for viruses and bacteria, so hopefully that will help inform future R&D spend in terms of crop protection for viruses and bacteria, and also for focusing biosecurity preparedness. We did quite a few disease identification workshops, which were fantastic, and unfortunately we wanted to run these nationally, but because of COVID we weren't able to do very many outside of Queensland. We did manage to get one done in Mildura a couple of months ago. These were really well received, and Hort Innovation's really interested in taking this further and making it its own sort of project. So hopefully that'll get up, because I think it's got a lot of value in both teaching how to identify diseases, but also as an opportunity to talk about new management strategies as they come through. We've developed fact sheets, and there's 9 of these already on the Hort Innovation website, and we will put them on Soil Wealth's website if they're happy to have them. And we've still got, so these are the ones that are available now. We did have a month to write some more of these. We actually only now have a week. But if you have any suggested topics and you want more fact sheets on anything, please let us know and we can write them up, and I'm sure Carl will help us get the information out. So biosecurity preparedness, we did contingency plans for 3 viruses. This is the begamoviruses, which are transmitted by silverleaf whitefly. Fortunately, we don't have very many of these viruses in Australia so far. We don't want any more because they're highly devastating. There's some tospoviruses and tabamos that we already have in Australia, but there's also quite a lot overseas. So that's thrips transmitted, and the tabamoviruses are seed and mechanical transmitted. We've done 3 contingency plans for bacteria. So bacterial wilt of cucumber, bacterial blight of onion, and Stewart's wilt of corn. And so these are all available through Plant Health Australia. Another big section was the diagnostic assays, and we've actually circulated 23 protocols through the national diagnostic groups to make sure that they're validated and accurate. 16 were for viruses and 7 for bacteria. And this used a whole range of different types of technology. New South Wales and WA in particular have been working hard to improve MALDI-TOF and BioLog for bacterial identifications. And collectively, all of these improvements mean that when you send samples into diagnostic services, you'll get an answer quicker, and potentially some of them you can do yourself. So we have 4 PhD students. Bianca has been working on phytoplasmas and has done a great job in improving what we know about phytoplasmas. Traditionally, we don't get very much money to work on them, and they turn up every now and then, cause trouble, and then move on. So it's good to have some money available to further understand what the phytoplasmas do. Joe's worked on developing new diagnostics for a range of vegetable crops, and a lot of this work's being done on cucumber, green CGMNV. And then we have Omar down at Tassie, who's just submitted he's probably the earliest. He's put his PhD thesis in for review, so hopefully he'll be awarded that soon, and has done a lot of work on filaroviruses in legumes. And finally, Noel, last but not least, Noel's been based with us in Brisbane, and he's been working on a Pseudomonas disease in zucchini, which had bad outcomes for growers in Bundaberg a few years ago. And so if you're looking for employees, there's 4 very good people coming out soon. So what's next? So there are some areas we've identified through the project that we believe need more work that we weren't able to do in the current project. It was a massive project, so it wasn't possible to do everything. The first one is virus diseases which affect lettuce and brassica crops, and these are both aphid and thrips transmitted viruses. And this has emerged as a big problem, particularly in the Lockyer Valley in Queensland and in several growing areas in Victoria, but also in Perth and Sydney Basin. And we think that a lot of the work that we've done in similar viruses in capsicums and zucchini could be easily transferred into the lettuce and the brassica crops. Another area is new age bactericides. So I think everybody accepts that the protectants that are available for bacterial diseases aren't effective under high pressure. And protectants will never be effective under high pressure. So we think we need a new way to think about bactericides, and we want products that actually reduce the bacteria once they're infecting a plant. And we've identified a few products through this project and some earlier projects that I think would work quite effectively to do this. So that would be another really good area to work on. And the last one is pectobacterium disease management. So during this project, we've seen pectobacterium diseases emerge as a real problem in particularly zucchini and brassicas. I think it's been misidentified for a while, and it's not known. The impacts have been not well recorded, and there's not much known on how the bacteria infects, how it spreads. So it's really hard to advise on how to manage the disease when you don't understand these basic processes. So some work along pectobacterium, I think, would be really beneficial for the industry. So if you think that these are good topics for further work, what can you do? You could talk to Hort Innovation about them and talk to your local vegetable science industry member if you think that you would like to see some work done on them. And that's it. I'd just like to thank, again, Growers the Agronomist, VegNet have been great as well, AusVeg, and of course Hort Innovation and the vegetable industry for the funding. Thank you.
Thank you, Cherie. Excellent overview of what has been an enormous project and a huge amount of effort that has gone into both the research and resources for industry. I might just quickly add too, Cherie mentioned that there was the opportunity to register for a hard copy guide or putting forward some additional topics for resources. Feel free to put those either in the question and comment function of the webinar or drop us a line in the comments section of the survey that we'll launch after this webinar, and we'll make sure we follow up with you and add those details in. So our next speaker is Paul Campbell, also from the Queensland Department of Agriculture, and Cherie mentioned virus, disease, outbreaks and weather. And we're going to hear a bit more about that in a deep dive now with Paul. So thank you, Paul.
Thank you, Carl. All good?
Great, we can see those slides and you're good to go.
Okay, so as I said, it's a quick talk on virus, disease, outbreaks and weather. Go to the next one. So viruses are quite cyclic. They're usually not a big problem until they are. So you get some years that you get smashed by a virus and then you don't see it for a decade, and then it comes back again and it wipes you out. Sometimes it just trickles on, but usually you see big boom and bust cycles for viruses. So what changes, what drives this? Sometimes it's a new virus entering the area, interacting with viruses that are already there. Sometimes it's a new vector that moves in, it's a little bit more efficient to transmit the viruses. Sometimes you change cultivars and it's a bit more susceptible. Usually, hopefully we're going the other way, that you're adding more resistances. And sometimes new weeds move into the area, which are an alternative host for the virus as well. Or the weather is changing it, and hence the topic of this talk. So I mainly focus on two little case studies that we are doing from this area-wide management project and the previous one, as well as another bean project through Hort Innovation. So tomato yellow leaf curl arrived in Bowen in about 2011, and it was the first time seen. It was a big jump into the area, so it wasn't a slow move. So it was a really good case study to see what happens when a virus moves into this area. In the first main season after it got in there, it caused losses of about $9 million. It's transmitted by a whitefly, and there's been occasional years where there's been massive outbreaks ever since then. Luckily, a lot of the growers have moved to tolerant lines, so the virus still kicks around in the plants, but you're not causing major symptoms. And the other one we want to look at is a disease outbreak in beans in the Fast Fern Valley. I first really noticed in 2016 autumn season, and it caused a large amount of wipe out of that crop basically. Beans pods were all twisted, couldn't be sold. And it was a whitefly transmitted Kala virus. So some basic virology. Primary spread is where the virus is moved in via a vector. So an insect vector moves the virus from weeds into a crop plant. Secondary spread is where it's moved within the crop itself. So the virus grows up in your crop plant and then the insects spread it to subsequent plants up and down the rows. Usually in Australia, we're looking at mainly primary spread and probably continual primary spread into the crops. So if you see secondary spread, you'll see some highly affected young plants and then ones affected about 2 or 3 weeks later, and then more affected another 2 or 3 weeks after that. That's when the secondary spread is going in and moving around. But if you see a big wave and all the plants about the same age, that's a primary spread. And because we spray for insecticides in the crop itself, we're actually not getting much secondary spread. It's just coming in from the outside continuously from those weeds. And how does the weather affect that? Well, insects don't like rain, or they do like rain, depending on the insect. Warmer weather can make the insects go through growth cycles a lot quicker, so you have a larger number of insects available to spread the virus. The virus itself replicates quicker when the weather's nicer, a bit warmer, replicates even quicker. But it's mainly the effect of the amount of weeds that are available to spread the virus into the crops. So let's have a look at this amazing graph that I have absolutely no idea what it means. At the beginning, so basically the numbers of whiteflies are all over the place. Some years you have huge numbers of whiteflies in Bowen, but there was no virus. Then there's no whiteflies and huge amounts of virus. And we also check whether the whitefly actually had the virus. And there's almost no correlation in this between the number of whitefly that have the virus and the amount of virus in the crop. It's just all over the place. It's the dry tropics. So if you get a lot of rainfall, pretty much November through to April, and then it drops off to nearly nothing. So if you look at, in this graph, they have the amount of whitefly versus rainfall. And so the year like 2014, 2015, over December, the rainfall dropped off early. So it was done by April, pretty much. And you end up with a large number of aphids. So in 2016, we had the reverse of that. It was done pretty much by April, but then we had and the whiteflies kicked off really strongly in the beginning of 2016. But it rained a lot in June and they basically wiped the whitefly population out. So in 15, we had a lot of virus outbreak, and 16, we basically had none because sure, the whitefly got off to a good start, but then it wiped them out in the weeds because they really aren't good in the rain. And we had quite a low virus year that year. The same thing's happening this year, like in tospoviruses. We're getting quite a lot of infection happening because of the weather. So I'll go to French bean. So 2 years we had lots of cowpea mold model in the French bean. 2 years we didn't. Temperature was pretty much the same. The summer and early autumn rainfall did vary, and that's what affects it. So high incidence, we actually had low rainfall, and low incidence where we had high rainfall. So the rainfall was just affecting the whitefly numbers. So the high rainfall meant that they didn't survive and actually be able to transmit the virus into the crops. There's a graph that just shows a bunch of lines that I'm not really sure what's about, but it's, again, the whitefly numbers peak early and then you get a lot of virus. Then pretty much you get almost no whitefly in the crops themselves, but the virus incidence stays high, and that's just the virus getting transferred into the crop from the weeds. And here's the rainfall. So those two high March, so they're autumn things, that's when we got the low incidence, and the low 2015, 2017, the low March rainfall is when we got high incidences. So what does this all mean? So usually late rain in an area that has no, that usually doesn't have rain, allows a green bridge into the early crops. So in the Bowen area and a lot of other cropping areas, the weeds grow up over the wet season or the off season. Usually there's maybe a month break, maybe a bit more, where the weeds die off because there's limited rainfall before the first crops go in. And so if there's late rainfall, it allows those weeds to growing all the way up until the first plantings come in, and then they start to die off, and they've supported a fairly large insect load, and they all move into your crop and spread the weeds. And sometimes, like I think this year, because we've had that just continual, sometimes very heavy rain, but continual rain throughout the season, the capsicum corosis levels in the Gumloo area are massive this year because those weeds are just still around to transmit the virus into the crops continuously. The virus levels are very dependent on, can be very almost hyper-local climate effects. So, you know, rain's not everywhere. It goes down one paddock and not in the other, and it can just support the weeds in there rather than elsewhere, or it can drown the plants. So again, whiteflies can't swim very well, which is good if you get quite heavy late rainfall. It tends to knock that population right down. And it's another reason to complain about the weather. So lots of thanks to everyone. Cherie's probably covered this already in the area-wide overview. Thank you.
Thanks, Paul, and some really interesting insights, I think, particularly against the backdrop of the reasonably wet and cool season that we've seen in the last couple of months, particularly the eastern seaboard of Australia as well. Just a reminder too, if you do have questions throughout these presentations, to please pop them into the question pane there and send them through, and we'll make sure they get to our panel discussion later in this session. I'd now like to hand over to Rebecca Roach, who's going to talk to us about pectobacterium in zucchini. Thanks, Beck.
Thank you. Hi, I'm Beck. I'm going to talk to you about, pectobacterium soft rot diseases in zucchini. So this is an issue we've been seeing crop up a bit in recent years in zucchini fields. This is essentially what you're looking at, a wilting and then a sort of collapsing of the crown and a squishy sort of soft rot around the crown and sort of lower branch area. So basically, upon seeing all of this cropping up around the place, we first wanted to take an initial look at what was going on with the symptoms and epidemiology. So first up, we obviously wanted to identify the bacteria causing this disease. We also wanted to make sure that we had the symptoms consistent and that we could replicate it for whatever studies we were doing. We also want to know how it spreads throughout the field, because that's quite important when it comes to any disease, but especially bacterial disease. So initially we were thinking, you know, is wounding with tools at fruit set and picking an issue? Insects maybe moving it, that can happen in some bacterial diseases. Is it a problem in seed? Is it moving through water? Is it surviving in crop trash or in the soil? So we wanted to investigate this with some pot trials and field trials just to get a sense of what we were working with. So first of all, we carried this work out with some colleagues over in WA who are also seeing this disease. So in identifying the bacteria, they did great work with this using the MALDI-TOF and biolog technologies, as well as molecular qPCR. And so, yeah, we identified that it was pectobacterium. We're going with these identifications here, Pectobacterium brasiliensis. You get slightly different identifications sometimes based on what database you're using, which is another thing that we want to, you know, down the track improve with all these technologies. But we've got an initial identification for the bacterial isolates we're working with. So then we moved on to pot trials. We did one in Queensland here initially. We took some soil from one of the infected fields that had this pectobacterium soft rot disease. And so we used that infected soil and we used some potting mix. And we basically did a small pot test with some inoculum with pectobacterium and some without inoculum, just to see if it was a soil-borne disease issue. So we inoculated some with pectobacterium and we got really good symptom results with the potting mix and the inoculations. Surprisingly, I suppose, the field soil didn't give us much symptoms at all, even when we inoculated the plants. So we got really low symptom severity without the inoculum and with the potting mix and the field soil and with the inoculated field soil. So we figured out that we can replicate the symptoms pretty well. And as to whether it's soil-borne, this seems to suggest it's not much of an issue. But this was just one preliminary pot trial. So we moved on to some more pot trials, this time over in Western Australia with Dom and Craig. Theirs gave us some pretty similar results. Does contaminated soil result in disease symptoms? Didn't look like it. They also applied inoculum to the soil in this pot trial without wounding it directly in the plant, and that didn't give much disease. And they also wounded some of their seedlings, and they didn't get too much disease either. So they carried this out for 2 weeks, and they didn't get much symptoms at all. So we did a field trial back in Queensland because we want to know if the tools transfer inoculum. We wanted to know if drenching the roots, that's applying the pectobacterium to the soil, would result in disease. And we used the same stabbing method that we did in the pot trial to replicate the symptoms in the field. So this is a little layout of our field here. When testing the tool transfer, we inoculated 2 plants down the bottom of the row, and at fruiting, these were, wilting and infected. And I'd cut the fruit off of these and then just keep cutting up the row without cleaning my tools to see how far the disease would spread. So what we found is that tools didn't really seem to spread it that much. If it did, you would assume that you'd get some more severe symptoms closer to the inoculated plants, and possibly, symptoms and wilting sort of trailing off the further up you went and the less inoculum you had on your tools. That's not what we saw. We just saw it pop up almost randomly in a few plants. The drenching row, it also didn't really seem to result in symptoms. Stabbing does, that's a reliable method that does induce disease expression, which is good to know. So we looked for the pectobacterium throughout the field, and as long as it's coming up in stuff you've inoculated, that's to be expected. It also came up in our control row. So what we saw, which was a little unexpected, was essentially wilting plants with pectobacterium-like symptoms randomly spotted all throughout this field in plants that I did not inoculate or go anywhere near with a tool. So that was interesting. Back over in Western Australia, they did a variety and host range trial. So the questions we wanted to answer here are: all zucchini varieties susceptible? Are any related hosts susceptible? And are different pectobacterium isolates also pathogenic on these related hosts? So this was a ton of work, and it gave some pretty great results. Basically, all tested zucchinis were susceptible. So you can see here, these were all the zucchini varieties that were tested. In this table here, B equals browning. So that's sort of wilting and lesions up the stem and leaf. O equals ooze, so like from the crown area, you'll see it's sort of, yeah, an ooziness coming out. It's very soft and wet and clearly degrading tissue. And the W is a severe wilt. So you got some symptoms in all the zucchini varieties, and it looks like slightly less symptoms with this particular pectobacterium isolate, but the P. brasiliensis, yeah, all gave symptoms in zucchini. So down here with the alternate hosts, these are all the ones that were tested, various pumpkins, cucumbers, loofah, some carrot and tomato, and basically the Jap pumpkin, the butternut, the loofah, carrot and tomato did not show any symptoms. But yeah, the odd other cucurbit and some melons did. They gave some browning symptoms here, which is good to know if you're looking to rotate crops or anything. So we also did another field trial back in Queensland. With this one, we wanted to know if we could track the inoculated bacteria through the field and maybe find out if insects are involved in the spread. Because as I mentioned before, having the disease pop up in our control rows was interesting, unexpected, and kind of an issue when you're looking to investigate how this thing works. So the idea is the pectobacterium that I've inoculated spreading throughout the field somehow, or is there just natural native pectobacterium that's, you know, in the seed, in the soil, in something, just popping up in the experiment and interfering with what I've inoculated. So we tracked it using a rifampicin mutant. Basically, it's just a sort of screening tool that I can use to make sure that the bacteria that I've put into the field is the same one I'm getting out. And that actually seemed to work quite well. This field trial resulted in much more rapid death than the first one did. The only difference I could think of was the prolonged rain period we had around April, May this year. The whole field was not inoculated, but the whole field died very quickly. And it sort of looked like most of it was this soft rot kind of wilting. So yeah, of all the things I inoculated, I managed to get this mutant out. So the tracking worked well. I also got pectobacterium that wasn't this, the one I put in, out of plants that I had not inoculated. So it seems like there's no evidence currently that the inoculated bacteria is moving through the field. It might in fact be that we have evidence of naturally occurring disease popping up in the control plants. So the next steps basically is to fill in all the gaps. We've done some pretty good work with the initial trials trying to figure out what this is and how it's spreading. We probably want to determine if it's seed-borne, because that's what with it showing up in controls, well, that's a pretty natural suspicion to have, so we'd want to check that out. We also want to determine the optimal temperature and conditions for this disease. With that rain event having such a devastating effect on the field, we definitely want to know if that's a major factor. We also want to investigate insect involvement. Unfortunately, most of our insect traps in that last field trial didn't turn up much of anything. They caught a lot of mud, and I suspect the magpies had a good go at them because they were fairly torn. So we'll try that again. We also want to investigate more variety and host tolerance, particularly in stuff that's more common in Queensland, just to catch up with the work that the Western Australians have done. And we also want to have a look at this emerging disease in other crops and regions. For example, we know that womboks get this too. Pectobacterium as a genus of bacteria has a lot of species within it that infect many, many things. They have an incredibly wide host range, so it's definitely worth checking out. So currently what we can recommend is that infecting through soil and crop trash is probably not a huge concern. And none of our tests really showed that it resulted in any kind of intense disease severity. So yeah, apart from that, thanks to everyone in the team. Thanks in particular to the WA team, Dom and Craig, for helping us out with all of this. Thanks to all the funding bodies and everyone. And yeah, that's us. Thank you very much.
Excellent, thanks Beck, and thank you to Cherie and Paul as well for delivering those presentations. It's now over to you, our audience, to put your burning questions to our presenters and get those answered. I'd just like to kick things off. We've had a question come through around monitoring and the importance of that. Cherie, I was hoping that you could talk to, in the first instance, its usefulness around controlling and properly managing disease as well.
Sure. That was on monitoring, Carl. Is that the question?
Correct.
Yeah. So we strongly encourage people to monitor for both the insect pests and the diseases. Monitoring does a couple of things. It gives you an early warning on what might be around. It gives you an opportunity to get samples tested early in the season, so you know what pest and pathogen that you're working with. And then over time, if you regularly record the data and you do it in a structured way, it gives you a build up over time of when diseases are likely to happen, where on the property they might turn up. So it can give you an indication of hotspots on a property where are more likely to have disease or not. And this applies to other diseases as well, like soil-borne diseases, as much as insect-vectored ones. For example, you might find that a certain block often has tomato spotted wilt turn up at a certain time. And if you look at that more closely, you might find the weed hosts next to that block, and then you understand that weed host is something that you need to manage and remove before you put your capsicum crops in that block. So that's an example of why monitoring is useful. And it goes hand in hand with good record keeping and also diagnostics. So we know that a lot of the viruses look similar, and not just viruses, even fungal and bacterial diseases look very similar as well. So having diagnostics done regularly gives you confidence, you know what you're managing.
Absolutely, thanks Cherie. And Paul, Cherie mentioned the importance of record keeping there too. I imagine also, just based on what you've just presented, a record of the weather conditions is also useful to track over time. Is that correct?
Yes, yeah, of course. And it helps if you can get like local data. So we're having to pull it from your bureau sites, which may be 2 or 3 kilometres away from where we actually are. The one from the FastFern was right there, so it's pretty good. But, a lot of farmers have, fields scattered all over the place now, and you can't rely on just one central weather data to, predict for the entire field. So, yeah, definitely keeping weather records and what happens during those weather events is a really good idea.
Absolutely, thanks Paul. And just while we're with you, Paul, too, we've had a question come in just about insecticides and how effective they are for insect control. I was wondering if you could comment on that.
For virus control, insecticides are not useful at all, unless you feel like spraying the weeds around your crop. Because of that primary spread events that are just continually coming in, the insects aren't growing up on the crops, and they're not, you spray them once or twice for insects, that's enough to knock out those populations, and they're not going to be spreading those viruses around inside the crops. So in the case of aphids, it takes less than 30 seconds of feeding to transmit viruses usually. So you're not going to be able to spray them out before they can do that. And occasionally, depending on the control chemical, you can actually, if they're not feeding properly, they're actually spreading the virus further. So they go, oh, I can't eat from this plant, I'll move to another one. And they actually spread it to more plants than they would have normally if you're not spraying for anything. So yeah, as long as you're keeping the insects under control, you know, because they can be a pest in themselves, you're not going to affect the virus incidence in your crops by insecticide usage.
Thanks, Paul. And an important point there, not only not effective, but can actually have the unintended consequence of spreading further, as you said, with feeding patterns and damage. So just while we're on control, Beck, I might come to you now just around bacteria control and the role of copper. We've had a question come in saying, should copper be sprayed for bacteria control?
Sure. So in terms of pectobacterium, it's not really going to help you out. Especially as like the pectobacterium is a sort of internal rot systemic disease. So the idea with copper is it comes in contact with bacterial cells and kills them there. So if it's like if it was a soil-borne or even seed-borne sort of disease, yeah, it's probably not going to help you much. In general, copper's not really going to help you that much. Its efficacy can be affected by a number of different things. The mixtures and stuff really affect how the copper actually works. It's generally it's why we're looking into alternatives. It's not really a great one, especially if you're just spraying constantly copper everywhere, because bacteria actually build up resistance to it really quickly. That's another issue we've seen for quite a while now, but it's becoming pretty clear that it's just not going to be much use as you go to anymore, and is one of the reasons we're looking into alternatives. So not a huge help to you there, unfortunately.
Thanks, Beck. Cherie, you mentioned the free diagnostic service for industry that was run during the current project. We've just had one question from one of our participants saying, is there the potential for that to continue into some of those future investments and RFPs with Hort Innovation that you flagged during your presentation?
Yeah, so the current project subsidised diagnostics for the veggies, and I can't comment. I guess that's up to Hort Innovation to I guess develop the scope of the RFPs for any future work. But I think it is a good idea to start embedding some of that diagnostics into projects as we go. I think it's a great way of again, monitoring diseases over time for the industry, because in our service, and I'm sure the other states are the same, we record you know, the samples that come in, and so we can build a profile of, you know, we can run a report to see what the most common pathogen that's come in on capsicums this year is, and where did it come in from, how bad is it, you know, and all that sort of information is really valuable for the industry on top of the actual free diagnostic that the growers receive. I think our state agencies used to provide diagnostics for free in the past as a general thing, but nobody's in a position anymore to be able to just do that. So it has to be subsidised through projects.
Thanks, Cherie. Beck, in addition to your response about copper just before, another question's come in off the back of that around a comment on the effectiveness of insect sprays to control bacterial disease.
So there's not too many bacterial diseases that are insect vectored. I looked for the insects because there are some, particularly in cucurbit, there's an exotic one that's vectored by a beetle. So, you know, maybe if you have insects in your field, you know, they can put their little foot in a droplet of water that's bacteria on it, and maybe, you know, wandering through the crop, spread it that way. But yeah, generally insecticides, yeah, not going to help you out too much. In terms of insects, I think just keep an eye on what you've generally got in the crop, and if you notice something new, I mean, that's cause in itself to get it checked out. But yeah, there's only a few, or not like as many diseases that are properly vectored. It's more of an issue for viruses.
Thanks, Beck. And Paul, your presentation highlighted the importance of weed management. I'm just wondering if you could make a comment about what that looks like in practice and where to prioritise or start if you're a grower around weed control and some of the issues that you've raised around that primary transmission.
Yeah, that's a big box to unpack. So a quick example is I originally started my PhD on lettuce necrotic yellows virus, and it was a big problem, and then it basically disappeared for a decade because the local council started slashing the roadsides better, and the weed, which was the main host, basically disappeared. Like, it was there, but not enough to host aphids. So it disappeared for over a decade just because of, you know, better roadside slashing of those weeds. It's coming back a lot now because people are leaving more fields fallow, and the areas on weed banks is, you know, the weeds build up on that. And that's probably where we're seeing a lot of these, like the bean one. It mainly comes out of weeds next to watercourses, or basically just on the edges of the field. They're not flying in usually from kilometres away. They're actually just from the field next door. And especially as your season goes on, if you have a little bit of virus in your first crop and you walk away, you know, we're trying to say spray it down before you mow it, maybe hit it with insecticide after it's done. That's an expense, but you will stop those insects moving out of that crop straight into your next one. So it's just basically looking around and seeing where those possible areas could be. Like Cherie was talking about the monitoring. If you see a lot of problems in that one field, maybe look at the areas around that and maybe see if you can slash those weeds down.
Carl,
Yeah,
can I just add a few things to what Paul's just said? And in terms of area-wide management, Paul highlighted a really important thing about the council. And, you know, if you're gonna, it's not just on property, and it's certainly not one block. You can't just manage these things on a single block. You do need to think more broadly than that. And yeah, it's the council does have a role in this, and I actually think there's a project there that Hort Innovation maybe should consider in terms of an area-wide strategy for reduction of weeds in a district. And it's important to note that if you want to apply insecticides to weeds, you really have to check APVMA guidelines, because it's actually you know, products are registered, and we have to be really careful what we spray in areas, particularly riparian areas. So what Paul said is absolutely true, and we do say this, that, you know, the best chance you have at stopping virus diseases is to stop those insects coming off the weeds, but there's a massive challenge there on how you do it. And I'd really like to see Hort Innovation, it's not one of the recommendations out of our project, but maybe it should be, but you know, that to look at a strategy for area-wide weed control. Yeah, that's all I wanted to say.
Thanks, Cherie. A really good follow-up to Paul's response as well. Just before we finish up our Q&A panel, I guess I'd be interested in all three of you commenting on some of the key takeaways or tips for growers in this area-wide management of disease approach. Where do you start? What are your top 2 or 3 takeaways? Cherie, we might start with you and then move to Paul and Beck.
I guess my top takeaway would be to consider the area more broadly. So if you have a disease on a block, first of all, get it ID'd so you know what it is, and that will inform on where it's come from. And then look more broadly than that block to see where the potential source for that disease is, and look at how you can manage it. And it is hard if it's you know, weeds in a neighbour's property. That becomes very tricky. But that would be my take home, would be get it ID'd so you know what it is and what's spreading it, and then look to see where it might be coming from.
Okay, great. Paul, did you want to add to that?
Not really. Know what it is, know where it's coming from is pretty much it.
Yeah,
That's
great.
the main things for the management.
Yeah, so I guess in terms of bacteria, my main takeaway for all of the bacterial work we've done is copper is maybe not the be all and end all. I don't know if it ever was, to be honest, but it's certainly looking like it's not going to be in the future. And yeah, we definitely need to try and find some alternatives. And as for, yeah, the management of bacteria, they're another one that tend to, you know, just linger at a sort of low level. Maybe you're not too concerned, but then as soon as the conditions are right, the weather turns a bit, you get some moisture, yeah, they go off really quickly. So definitely keeping an eye on what you've got just around in general, and then definitely monitoring the weather and trying to factor that into yeah, how you grow, and just looking at your crops for what you've got. Yeah, that's pretty much it, I think.
Thanks, Beck. So we've got know what you're dealing with, correctly identify the problem, know where it's coming from in terms of source and perhaps your neighbours or the roadside, and then a couple of specific ones there around bacteria that, you know, copper might not be the silver bullet, but the importance of monitoring too, I think, across those things. Look, thank you very much for your questions and for sending those in. We're almost at the hour now, but before we do finish up, I just wanted to signpost to and events available through Soil Wealth and ICP. And as Cherie mentioned earlier in her presentation, we'll have those guides and fact sheets available on the Soil Wealth website, which is up on your screen at the moment. But there's also a host of other things like best practice guides and demo site reports, podcasts and videos there for you to watch and read at your leisure. We're also planning a 3-part mini webinar series with the research team from the Area-Wide Management Project, kicking off in August and running through to October. And we're going to be covering specific viruses in those of curcubits, capsicum and lettuce with the teams from Western Australia, Queensland and Victoria as well. So please jump onto the website and register for those if you haven't already. And we look forward to seeing you at the next event. I'd also like to thank all our presenters today, Cherie Gambley, Paul Campbell and Rebecca Roach, for their time and insights and expertise. We really appreciate that. And don't forget to register for your interest in the hard copy guide and any additional topics for those fact sheets. I've been Carl Larson from RMCG, and we look forward to seeing you at the next session. Thanks, everybody.